Remember that on your revision you can use basically any resources that you want other than professors this semester or last semester, UTFs or SIs. Okay? None of the teaching group. Yes, question?
So when you say different version of the test, will it be the same questions or the same types of questions?
The second version will be the same questions. The only difference is when you take it the first time, the answers are randomized. Okay, and in the second time, you will all have exactly the same version with the, they'll be maybe in a different order than the first time you took it, but everybody will be doing exactly the same thing the second time. All right, so I think one of the best ways to approach this second version is to get together with other members of this class, right, and discuss them, go through each question. Hopefully you kind of remember what you chose, decide whether you want to keep that or if you want to make some changes. Okay? Yes?
Is there a time limit on it?
There is a time limit. You have to have it turned in and submitted by Friday night at 11:59 p.m. In the meantime, you can go into the exam. You can go back out, you can go back in until you actually tell it to submit.
Cool. Thanks.
Then you're done. And remember that if you're going back in and back and forth, before Friday at 11:59 p.m., make sure that you do submit it. Okay? All right? Any other questions? Yeah, question.
Once we submit it, when will we be able to see what are the correct answers are?
You'll be able to see the answers after the deadline. Okay, because I don't want to open up any of those while other people are still working on it. Okay? Any other questions? Yeah.
So when will we get our average scores back? So
after the deadline. Right? So I can, and then I'll have to go into Canvas and probably pull it back out into Excel and do averages and re-upload it. And the total score, remember the total score for the exam will be the average of your 2 computer attempts plus the written portion. Right? And so all of that won't be done, obviously, until the written portion is done as well. Okay? Any other questions? All right. So partly because the exam is still going, we're going to take it kind of easy today. And what I'm going to do is just spend the day looking at something that won't ever be on an exam. Right? So you're probably thinking, oh, why am I even here? Oh, I could still be in bed or something like that. Right? But my point is that, what we've seen as far as NMR especially is just like the tip of the iceberg. And so my hope is that by showing you a few more advanced methods, it will at least trigger your imagination a little bit. There is more out there, and that can be interesting, I think. All right? So, all we're going to do is we're going to just kind of survey some advanced NMR techniques. All right? Tomorrow, we will start into Chapter 16. Okay, so Chapter 16 is on conjugation, resonance, and dynein. So we're going to start to talk about resonance. There will be some reactions that are related. Let's quiet down, please. There are some reactions that you've learned before, but if we put them into conjugated dynein systems, we can get a different outcome. It gets just another level of complexity. So then, after we do that, chapters 17 and 18 are about the chemistry of benzene. Okay, and the chemistry of benzene has a lot to do, so this is kind of an introductory chapter to those 2 chapters. All right, and as you maybe have noticed, I do have notes up for 16, but I'm trying to stay at least 1 chapter ahead of you. because all my notes are from an old textbook, so I have to revise everything as we go forward as well. Okay? All right, so let's look at some more. Okay, so just a little bit of history. NMR was first used, or the first spectrum was observed in 1945. Okay, so it's been a while, and several people, in fact, won the Nobel Prize for observing the first NMR spectrum. So it was a big deal. So in the beginning, the magnets, though, were like solid iron magnets, right? And so the way that the experiments were run were either they could scan through the spectrum essentially one time, and they had to either vary the magnetic field or they had to vary the radio frequency, and they would just like vary through the radio frequency, and when the radio frequency and the magnetic field were the right combination, they would get a peak. Okay, but it was very insensitive. So in 1964, still pretty long ago, computers even then got to the point where they could do what's called a Fourier transform. And so this is a mathematical process where they can convert a data that is recorded based on time, and they can convert that to frequencies. So they record a bunch of frequencies, and those are the frequencies that correspond to the energy gaps for these different protons, but they can convert those frequencies or the time into frequencies. And so this allowed experimenters to not only scan through once, but you can do all the frequencies at once and then separate them with this Fourier transform. So you can scan over and over again, and each time you do that, the positive signals continue to add, and the noise is random and it subtracts. Okay, and so this gave much better signal-to-noise ratios, made it a much more sensitive technique, and so you could do it with smaller amounts of material, more reasonable amounts. And so there was another Nobel Prize in 1991 that was awarded for this experiment back in '64. In 1974, the first, what I'll show you, is called a two-dimensional NMR experiment was devised and run. And then in the 1990s, they started to apply this not only to small molecules like we've been looking at, but to proteins and large molecules. It gets very complicated. And another Nobel Prize was awarded for that. Okay, so you can see by the number of Nobel Prizes that have been awarded just for doing NMR spectroscopy that this has been just a revolutionary technique in chemistry. So we talked about this a little bit before, the chemical shift equivalence, but I was just going to bring this up again as we look at some more complicated spectra. So if we have molecules that have stereogenic centers in them, like this center at number 4, okay, so that's a chiral center. The protons on the adjacent CH2 groups have different environments. We call them diastereotopic. So in this molecule, whereas if we had more simplified version, we would think that you'd have one signal for the protons at carbon 2, one signal at 3, one at 4, 5, and 6. So we'd think we'd have 1, 2, 3, 4, 5 signals, right? But we actually observe 1, 2, 3, 4, 5, 6, 7 signals. Okay, so it's more complicated than what you would think. And the reason is that the protons at position 2, the protons at position 3, and the protons at 3, that's not exactly, yeah, at position 5 all of these are diastereotopic. Okay? So these are kind of labeled already. So here's the 2 protons at carbon 5. And notice that it's going to be much more complicated then, because those 2 protons at 5 will couple with each other because they're non-equivalent, and they'll couple with protons on adjacent carbons. So there's the 2 for 5. Here's the 2 for 3. And notice how far those are apart. The protons at 2 are actually still overlapping. Okay, so this provides us, in fact, it can provide us with some additional information. Okay, so here's this. Oh, I got rid of that one. So let's look at a more complicated spectrum, though. So this is a spectrum, a proton NMR spectrum of this compound called cinchonidine. And these spectra were all run, I did them in fact, in one of the NMRs that we had in the basement of the chemistry building. And so notice that there's just a lot of signals, right? But we have really the same things that we could use before, right? Look at, so first off, we might look at chemical shift, right? We know that at one end that I have boxed in blue and drawn in blue on the structure is the aromatic range, right? So we know when we get down to maybe 7 parts per million down here, this is all the aromatic protons. We know that if we go to the upfield side here, this is more the carbons that have the protons that are attached to sp3 hybridized carbons, right? But we got a bunch of them. And then in the middle here, we actually have some, we have the protons that go on this double bond up here, and we have a proton that is next to an alcohol. Okay, it's still in that same region. It's next to the aromatic ring, and so it moves a little farther downfield. So, you know, we can look at general chemical shift ranges, and that first tells us something. Okay, we can look at the integration. So if you can see these, there are integration curves on all of these as we go through here. And so we can see these integrations, we could estimate these as most of them are 1 and some are 2. Here's 1 for 3. But some of them that are 2 are kind of overlapping signals, and they're not always equivalent, right? So we got this is a pretty good spectrum because a lot of signals are spread out, but there's still some overlap as well. And what we'd like to get some information is about the splitting, okay? And the splitting is a little hard to see in this one because all these peaks are kind of compressed. But if we could look in close, these out here, this one and this one might be a couple of doublets, right? And what else could we pick out? Nah, the rest is pretty complicated to pick out. So what do you do then if, well, okay, I'll get to that in a second. So another part of this is that when we looked at some of those spectra, notice that even though these are doublets, they're not exactly 1-to-1. Okay, one peak is higher than the other, and maybe you've observed this sometimes in the spectra that you've looked at. They don't end up at the same intensities that we expect, you know, the 1-to-1 or the 1-to-2-to-1 or the 1-to-3-to-3-to-1, because there can be something that's called second order effects that take place. Okay, here's a few what we would call first order coupling. These are more what we would expect, right? So here's 2 ethyl group, you know, this is symmetrical but an ethyl group, a quartet and a triplet. Here's 2 CH2s, and so we see a triplet and a triplet. Here's 2 H's like this, and so they're just split by each other, so there's basically 2 doublets, though notice these are slightly tipped a little bit. All right? So what in the spectrum is that if if two protons are coupled with each other, if their distance apart from one another in chemical shift is very far, then we get what we call the first order coupling. And that's what we predicted from Pascal's triangle. However, as the chemical shift difference gets small and on the same order as the size of that J value, the coupling constant, then we get what we call second order effects. So these values here, this delta delta is the difference in chemical shift divided by the coupling constant. And as we get close, notice that the middle peak of these doublets starts to get bigger than the one that's on the outside, right? And the way I always think about this is that I kind of draw an arrow from the small side to the big side, like this. And when we have this sloping, it tells us that, say, this proton here is coupling to a proton on the side where the arrow is going. Okay, so they're just tipped a little bit. It's becoming second order. If we make this ratio, they're both in Hertz, down to 1.5, look at it, now looks like a quartet, but what it really is two doublets that are just very second order. As they get even closer, the outside peaks get really very small. It almost looks just like a single doublet, but those outside ones get small, and if the difference in chemical shift is 0, then those are essentially equivalent now, and there's no coupling anymore. Okay? So these second order effects closed on what we might see. So C13 is also useful, but it's still insensitive, right? Though it's good because it has a much wider chemical shift range, so it's easier to get peaks that aren't overlapping on one another. We usually do it decoupled, which is what I showed you before. So they're all singlets. This is the solvent right here. And one way that we can get information about them is a different kind of experiment called a depth experiment. So what a depth experiment is it's a C13 NMR experiment, C13 NMR. But it's different from the normal one in that it only shows protonated carbons. So only carbons that have protons on them. Only protonated carbons. And we can essentially separate the signals based upon how many protons they have attached to them. Okay, so if you do it in a fancy way, you can get one spectrum that's only CH3 groups, one spectrum that's only CH2s, one that's only CHs, and then you can tell which are the quaternary that have no H's if you compare those to the normal spectrum. Okay, another way that's kind of faster to run is that you can run it so all of the CH2 groups, all the carbons that have 2 hydrogens are pointing down, and all of the CH and CH3s are pointing up. Okay, so this is a way that we can get some information about how they would be split if we did a coupled spectrum. Okay, so this is useful information. What do we do if all of the protons are still overlapping? Okay, then we can do what we call two-dimensional NMR. All right, so this is an example of an experiment. It's called a COSY experiment. It stands for correlation spectroscopy. And so this is what I would call a homonuclear experiment. So it's coupling between 2 of the same type of nuclei. So these are coupling between protons. So the way that we view it, the middle part is essentially a contour plot. It's like a topographical map. So if we could blow this up, each of these dots has different contours, and that tells us how tall it is. Okay? The real one-dimensional spectrum in this experiment lies along the diagonal. So this is where the one-dimensional spectrum goes along this diagonal. Okay? Everything that's off the diagonal shows us coupling between different protons. Okay? And so the way that we observe the different couplings is to look for peaks that are symmetrically disposed across the diagonal. Okay? So let me show you. So for example, if we were going to try to pick out the coupling in these different systems right here. We start over here on this peak right here. That goes to this one here. This is in fact the OH peak. All right? From the OH peak, if we go horizontally or if we go vertically, we run into another little peak. Okay? And if we went horizontally, we hit that peak, and then we go up, we run into a peak under here, which is the proton on the carbon where that OH is attached. So this one right here. Okay? It turns out that that proton there, this signal right here, has a couple of different peaks that are overlapping. I think it's the taller one right here, which is the one that's next to the OH. All right, once we've gone to that point, we can continue. We can continue out this way. All right, we run into this peak here. And then we can continue back up this way. And we run into a peak that's here. That's the proton on the other side of the alcohol. Okay, so these off-diagonal peaks tell us where the coupling is. And so we can actually see coupling even if we have multiple peaks overlapped. We can see that there's a coupling to a proton underneath there. We don't have to pick out and measure J values. Okay, in red are the protons that are around that double bond. And so it's the you see the small peaks in here. The more complicated signal underneath is this proton right here. Right? Because that couples to both of the protons on the end and the one back to the other side. And so it couples, again, we could go from there, we'd go up and across, and we run into all of these signals, and these are the two protons that are on the end. Okay? And those are looking like that because, remember, the two protons on the end, they only couple with, these two protons only couple with this one. One of them is a cis coupling, which has a smaller coupling constant, like 10 hertz. The other one is trans, is more like 15 hertz. And so if we look at those signals up there, up here, we'd have one splitting, the bigger one, from this peak to the tall one would be the trans, and then from that tall one to the small one is the cis, and they're just overlapping on one of those peaks. All right, so the spectrum, here's a blow up, so you can expand them, look more carefully. Here's how it's going around the aromatic ring. So notice these are the two doublets that I labeled before, but now we can see them better. So if we start with one of those doublets, like right here, and we go up, we run into this peak, we go across, and we see that it couples to a peak here. And so that's going from this one to this one. We follow the blue up and across. That's going from this over to the next one. We come back down. We could go back, or we go down here and back over to here, and we get to the other doublet. and that's the proton on the other side. So we can just kind of walk our way around this benzene ring by using this same kind of coupling. If we look at this proton out here, this small doublet, that turns out to be the proton right here next to the nitrogen. Okay, that's this one here. So where do we find its coupling partner? Right, so we would go either up or across. We could start here, we could go across. We run into this off-diagonal peak, and then we go straight up like this. So this doublet should have another doublet for the adjacent proton that has the same kind of coupling, and so that's what we see sticking out of the top of that one. Okay? So we just walk our way around. And so this works really well with complicated molecules, and we have more overlap of signals. All right, we can also have what we call heteronuclear experiments. And so these experiments show coupling between protons and carbons. All right, so notice that I have a proton spectrum on this side and a C13 spectrum on this side. Okay, and this experiment, it's called HSQC, tells us which protons are directly attached to which carbons. Okay, it shows a one-bond coupling. Right, so for example, we could start with this proton here, which remember was the proton right here, and there's no diagonal in this case because the two axes are different, but we just go from here, we go down to this one, and we go across to this one, and we see that this proton here is connected to the carbon over here. Okay? And likewise, we can go all the way around. Notice the whole aromatic, let's see if I have a blow up here. An aromatic region down here, and there's a peak here for each of those protons, and they correspondingly go to the carbons. All right? We can run this also in another way. Here, let me show you in a second. But here, a couple of important things. Yeah, so we see the direct connections. We only see the carbons in here that have protons on them because they have to have this proton coupling. And the other thing that we can identify are those diastereotopic protons. So if we see two protons that go to the same carbon, that means that we have this diastereotopic CH2 group. So for example, I have some highlighted here in blue. Right? Notice that there's two different protons in this axis, and they both go to the same carbon. Right? Likewise, well, the other one, it just shows one. So here's the two diastereotopic protons, and then here's this proton next to the double bond is the linking that's shown in red. Okay? So we can also run this in a different way. That's kind of a combination of this depth spectrum. Remember that I said the CH2s could go down and the CHs and CH3s could go up, and this two-dimensional one. So I can run this in a way that's called phase edited or multiplicity edited. So think about what this looks like. Let's look, so identify this carbon and protons there. This goes to the CH2s on the end of the double bond. So that's a CH2 group. In the next spectrum, those are gone, right? Because all we're looking at is the CHs. Okay, so we only see peaks in this spectrum where we have one proton going to one carbon. And then we can run another spectrum that's only the CH2s, right? And so that's where this one shows up now. And here's the diastereotopic pair. There's another diastereotopic pair. And in fact, I think there's another one under here. All right, so we can really start to separate these things out, and we can detail. All right, we can go up one level. We can do another heteronuclear coupling experiment. This is called the HMBC. This is for the heteronuclear multiple bond correlation. They always come up with these fancy acronyms for NMR experiments. And so what this does is it also shows proton to carbon coupling, but through 2 or 3 bonds. Okay, so essentially what it does is it shows coupling from a proton like this to a carbon like that. We can see interactions like this. So that's through 2 bonds. Or it's very good if we have systems like this. I'm going to put a Z group in there. So let's say that Z is an oxygen, and there's no protons on it, so we can't see coupling past that. Or it's just a quaternary carbon. There's no H's on it, so we can't see any coupling past it. But this in HMBC experiment, we can see past that group and see which protons are connected to another carbon. And so this is very good at connecting together different pieces that you generated using, say, that COSY spectrum. All right, so notice that this one's actually much more complicated. And I don't know if I don't really have any specific ones. Maybe on the next slide to point out here. Yeah, I got some on this next one. Okay, so before we had kind of looked at these 4 protons in the COSY spectrum. We could see that they're all next to each other. We could see that these 2 protons on the other end were next to each other, but we couldn't see how those were related to one another, or how this group here was related to the benzene ring, or how that group was related to this other side. Right? We have separate pieces, we want to connect them all together. So, what we can do here, so right here in blue, this peak up here, remember, was the proton that's next to the alcohol. So that's this one right here. And if we just go down from there, we see a peak here, one here, and one here. And so those are all carbons, if we go across, that are either 2 or 3 bonds away. So the line in yellow shows the coupling between that proton to this carbon over here. Proton to that carbon. The one in red down here at the bottom is the 2 bond from that proton to this carbon. In blue, it's the proton to this carbon. Right? So we can clearly through this place this proton on a carbon next to this benzene ring right there. Okay? We can also connect it in the other way. So that same proton has couplings here that go in red. That's from this proton over to this other ring system. And in blue, it's from that proton all the way over to this other carbon over here. So we can, from all of these, we can see that this carbon with the alcohol on it is kind of a central portion, and we can see what pieces are attached to that. All right, another type of experiment that's called the nuclear Overhauser effect experiment. So Overhauser is somebody's name that invented this. Shows interactions not through the bonds, through coupling, but it can show interactions through space. Okay, so it shows peaks that indicate that protons are in close proximity to one another, even if they're not close in bonds. So we could have protons that are like, if the molecule kind of curled around and it was a lot of bonds apart, it wouldn't show any coupling. But if they're close together, then in this nuclear Overhauser effect, we can see that they're close together. Okay? This helps us to determine things like stereochemistry, because like these, it's kind of hard to see here, but the ones in yellow over here to the right are peaks that show that all three of these protons are in axial positions, because that's the only way that they would show these kinds of NOE signals over here. Right? And so if this is in an axial position, then that tells us that this whole side chain has to be equatorial. So it told us the relative stereochemistry of that position relative to these other ones. Likewise, we can see some NOEs down here off of these double bond protons, over to this side and over to this side. These are kind of sometimes far away, especially in red, far away to see coupling, but they're pushed close together. This kind of experiment is also used for determining structures in proteins and in things like DNA, because you have a helical structure. There's, every 4, I think, base pairs, it comes back in a circle, and they can see these NOE signals, and they can tell which ones are all on the same side of the helix. Okay, so just a couple of general things, so we know already that we're detecting nuclear spins. NMR tells us about the environment, the relative numbers of nuclei, the connectivities, the spatial arrangements. One thing we didn't talk about is that we usually run NMR spectra in special solvents. And those solvents all have the hydrogens replaced with deuterium. And the reason is because if you took, chloroform is a CHCl3. If you took just regular chloroform and you dissolved your sample in chloroform and you tried to run a proton NMR spectrum, the concentration of your analyte is way lower than the concentration of the bulk solvent. So the H from chloroform would be a huge peak, and all of the ones that you want to look at for your analyte would be tiny. Okay, so the way we get around that is that we use a solvent that has most of the protons for deuterium protons substituted for deuterium. So most all of the solvent is CdCl3, and deuterium doesn't show up in the normal proton spectrum. There's always a little bit of residual proton, CHCl3, in the CdCl3, and so we usually get a little small peak. All right? But we can use, chloroform, DMSO, this is deuterated methanol, deuterated acetonitrile, acetone, water. There's lots of deuterated solvents that you can use, and you kind of choose which one can dissolve your sample, and also how much it costs. Because for organic chemistry, deuterated chloroform is super cheap, and so if you can get away with that, that's the one you use most often. We talked about higher magnetic fields, makes the magnets more sensitive, and it separates the signals. So if we have a bunch of multiplets, they might be overlapping at a low magnetic field, but at a high magnetic field, it separates them better, and we can get better dispersion. And the last thing here is that these instruments are not cheap, and so like in our department, there's usually a kind of a shared multi-user facilities. So they have 1 or 2 or 3 big instruments, and everybody from the department or all across campus can come and use that instrument. It's not usually an instrument that one researcher has in their lab, unless they're like an NMR researcher, and then they might have their own. All right, so that's all I have to say. So hopefully you know, you can maybe imagine what you might have to do if you had a much more complicated molecule. Maybe you need more time than what I gave you on the exam, though. All right, so I'll see you guys tomorrow, and we'll start into the next chapter.